Well, welcome everyone. It's great to be here, and I see so many familiar faces. I've known for a really long time. It's wonderful, and then all kinds of new faces, which is just absolutely really great. And this is meant to be. This talk is really. I put a bunch of different things together. We're going to talk about telomeres. We're going to talk about genetics, and then we're going to give sort of a high-level overview of some of the things you all already know because you're here and you know what you're dealing with a telomere biology disorder or something related. So just to start, to kind of set the stage, is what are telomeres? So telomeres are, if we think of our tissues and then our cells, our chromosomes, which are the packages for our DNA is all coiled up into those chromosomes. But at the end of the chromosomes is a cap called the telomere, and it has to, it's really important to have it there because it protects our telomeres from degrading. You can imagine that if you had sort of something here at the end that's not kind of nicely packaged, nicely bound by proteins or something, that the telomere could be susceptible to degradation. And so telomeres are this DNA repeat. So I'm going to talk about DNA in a little bit more detail, but it's these nucleotide repeats. So TTAGG is how we refer to them, but it's this long repeat of thousands and thousands of these 5 base pair repeats, and also a complex of proteins that help to form that cap at the ends of our chromosomes. And this is an analogy that many of you may have seen, is that the telomere is like the end of the plastic cap of your shoelace, which until I was starting to work on telomeres, I did not know that it was called an aglet. That was a new vocabulary word. But it's basically, if you think of that plastic cap at the end of your shoelace, right, it keeps it from fraying but over time, if you have your shoes and your shoelaces long enough, that plastic cap can start kind of fraying, and then if it comes off, actually your shoelace gets shorter, right? Because it's not stable anymore. So we kind of think of telomeres as this protective complex of DNA and of proteins that help make our chromosomes stable. So, and telomeres are essential to protect our chromosomes, to protect our whole genome. And actually, everyone has them except for bacteria. Okay, so even pond scum, and actually they may depending on their genome. But anyway, between like this mushrooms and bees and chimps and pond scum, which is actually tetrahymena, which is where a whole bunch of foundational work in telomeres has been done. All of our cells have something to protect the ends of our chromosomes, okay? And why do they need to be protected? Because just normal everyday life, we're susceptible, our DNA is susceptible to damage, whether it's ultraviolet radiation, whether it's cell replication. So every time our cells divide, our telomeres get a little bit shorter and a little bit shorter. And that's because every time the cell divides, the DNA has to completely replicate itself. And at that end, that telomere end, the DNA replication machinery just can't do a good job of replicating that telomere end. So every time our cells divide, our telomeres get a little bit shorter and a little bit shorter, and that's just a normal part of aging. And I'll show you some graphs of that. It happens to everyone. But there are also other factors that can affect DNA stability and DNA damage, including chemical exposures, as I already said, ultraviolet light, even our metabolism. Some folks have shorter and longer telomeres. And telomere shortening is actually a good thing in most instances, okay? Because every time a cell divides, as I said, telomeres get shorter and shorter. And it's this protective mechanism because when telomeres reach a critically short length, the cells are supposed to die or undergo what we call senescence. And that's actually a good thing in most cases, not all, but in most cases, because our cells can accumulate more and more DNA damage with each cell division. And so you do want to kind of come to an ending point where you have that sort of protective mechanism. And this is just a schematic that shows why and the range of why telomere shortening happens, and also why it can actually be a good thing When we think about telomeres, and I'm not talking about telomere biology disorders at the moment, I'm just talking about telomere biology and cancer. So, as I said, every cell division, telomeres get a little bit shorter, a little bit shorter. When they reach this critically short length, they're supposed to undergo apoptosis, which is what we call programmed cell death. This happening to all of us right now, okay, in different tissues. Or they undergo what we call senescence, which means they don't replicate, they don't do a lot, but they're actually very pro-inflammatory, those cells. But then there's a couple different things that can happen if we think of the general population and moving on to cancer, where if they upregulate or increase the activity of certain genes, they can the cells can keep dividing despite having underlying genomic or DNA instability. And then these are cells, the chromosomes get fused, they can cause all kinds of problems and accumulate mutations that could set the cell on the way to cancer. And then at the other end is some cells will increase telomerase or TERT, which a lot of you have probably heard about. And actually at the cell level, increased telomerase, those cells can keep dividing when maybe they shouldn't be because they've accumulated mutations. So this is thinking about telomeres and cancer development in the general population. So it's different, it's a little different for TBDs, but this is kind of the basis of a lot of what we understand. And I want to emphasize too that maintaining telomeres is really a balancing act. Again, thinking general population, average, everyday people, we all have genetic variation, common and rare, that affects our telomere length. There are some environmental exposures that in the general population can cause small differences, but they can be statistically significant in telomere length. And also the method of measuring telomere lengths really matters. There's been a lot of work looking at different methods of telomere length that are, some are really good, like FlowFish is good for TBD diagnosis, but other methods that are more DNA based are good if you're studying thousands and thousands of individuals. Okay. So let's go back and introduce kind of DNA, and let's talk about that a little bit more. But I hope most of you or all of you have had genetic counseling of some sort in part of your testing, part of your evaluations. When I think of our DNA, right, it's in our cells, it's packaged into our chromosomes. And think of your DNA, your genome, as a whole book, right? It's a book that has the instructions for how to make everything that you need to live. The chapters in the books are the chromosomes, okay? We have 23 pairs of chromosomes. So if you think of this, our genome is a book that has 23 chapters, okay? But actually, we have 2 chromosomes, we have pairs, so we have 46 chapters, okay? And then if you think in the book, you have a sentence, and the sentence is the gene. So the gene is what basically gives the instructions to the cell to make certain proteins. And if you think of a book and reading things, you need a complete sentence to understand what to do next, right? In this example, it's the gray cat ran down the hall. Okay. So if a sentence is a gene, okay, our DNA is made up of these bases, A, T, G's, and C's, which are basically the alphabet, okay? So our DNA will tell, except instead of having 26 letters, we have 4 letters that make our genes, okay? So the gray cat ran down the hall, and then if there's a variation or a mutation, you can see if you change the H to a B, right? The gray cat ran down the ball. That changes the sentence, and it's almost kind of not totally sensible, but it kind of is. So a DNA change, just like changing one word in a sentence, can change what it means, what the gene means. So we all have lots of variation in our genetic code. Okay, everyone's DNA is different. It's what makes us all look different, act differently. And our variations in our DNA are what make us all unique. We all have thousands of common genetic variants, again, that combinations of genetic variants have made me taller than, say, Dr. Lucic. Pick on her. But they've also, right? I can say that because my kids are combinations of genetic variations have actually made my kids much taller than me. Okay. But other combinations have given us green eyes, brown eyes, black hair, blonde hair, right? All these different combinations. And that's what's really important, is that in the context of even having a rare genetic disorder, we still all have those same we have common variations that make us all different. And we're a lot like our parents, we're also a lot different. We're more like mom, we're more like dad, or some sort of combination. And that's because of common genetic variants, okay? And variations that cause a significant medical problem or developmental problem are usually called, in the past we've called them mutations, but really the terminology is changing a bit. We're calling them rare pathogenic, which means not good, or variants. So if you think of rare pathogenic variants as causing disease, and then the common benign variants we all have are just what make us all a little bit different. Okay. Some variations that also gets complicated, especially when we're talking about bone marrow failure, is that we can also acquire mutations or variants in our cells throughout our life. And that comes back to what I was talking about with telomere shortening with each cell division. And every time our cells divide, they have to completely replicate 3 billion base pairs of DNA. So errors happen. In the vast majority of the time, there's no problem, they're fine. But in our tissues, so if you look at a newborn child, a 40-year-old, an 80-year-old person, the older you get, the more likely you are to acquire what we call somatic variants or mutations. And we know those best in the blood, and that's becoming a big factor in understanding TBDs as well. So this is just a picture of chromosomes. So this happens to be a male, and you can see they're different sizes. We number them 1 to 22, and then X and Y. Okay. You can see that there are also pairs, and the darks are bands, which is part of the staining process. And the X and Y in the bottom right, so this is a male. Males have an X chromosome that they inherit from their mom, and then Y chromosome they inherit from their dad. Females have 2 X chromosomes. Okay, and that's important when we talk about inheritance, but this is basically what our chromosomes look like. So X-linked recessive, so we're going to talk about that in the context of TBDs, is a disease where you inherit a pathogenic variant from the mother, in this instance, who passes it on to a son, and only males are affected in this type of inheritance because males only have 1 X chromosome, right? They have an X chromosome and a Y chromosome. So if you have an abnormality on your X chromosome and you're a male, you don't have anything to protect you from that variant, okay? If you're a female, you have a second X chromosome. So in this example, even this female who has an abnormality on her X chromosome, chances are she's going to be fine because she has a second X that balances out that abnormality. Another form, though, is so that's just the sex chromosomes, X and Y, where we can have X-linked disease. Autosomal dominant, the autosomes are just the other 22 pairs of chromosomes. And if it's dominant, it means that you only need 1 variant or 1 mutation to cause disease. So in this example, the father has a mutation on 1 chromosome, not on the other. The mother doesn't have it. So it's actually every child they have is a 50-50 chance that the child will inherit this because and so he can pass on either this chromosome or that chromosome to his children, right? The mom doesn't have a mutation in this example, so she is not going to pass that mutation on. And each time, it's basically a 50-50 chance which chromosome they're going to inherit. And then males and females are affected equally because this is not on the sex chromosomes, okay? The other form of disease that we also see in the TBDs is what we call a new mutation or a new variant. This is where the parents are unaffected, as far as we know. We do all kinds of testing, we can't find the variant in them, but they have a child who has a rare variant in one of the telomere biology disorder genes. That usually we think happens sometimes spontaneously in the egg or the sperm that made that child. Okay. It's really difficult to track down, but we see that fairly frequently. We see that more often in the TNF2 gene than in some of the other TBD genes. And then autosomal recessive disease, basically you need two bad copies to have disease. Okay, so in this example, each parent is a carrier. Each parent has one abnormal gene. And each time when they have their children, there's a 1 in 4 chance that they're going to inherit, that this child is going to inherit this mutation and that mutation, right? Versus the other children could be carriers or not inherit it at all, okay? And this gets extra complicated in TBDs because that's just the way these disorders need to work. In some cases, you can have a parent who has the dominant disease who presents later in life. And then they can still have children who present with earlier onset disease because they have the two variants, the two mutations. And sometimes that's how the children are first picked up. So that's kind of a really quick overview of kind of genetics 101, and I'm happy to talk through more specifics at any time. I thought it would be nice to do a little bit of a history of TBDs to help put things in context, sort of why we're here. I think the background in telling the story helps understand how we've gotten here. So the first reports of dyskeratosis congenita, or DC, were actually around 1906, 1910. And they were reports of children, usually boys at this point, with abnormal skin pigmentation, abnormal nails, and bone marrow problems. And what they were describing is essentially this X-linked recessive pedigree, which was a family had known for a long time. You can see in this case, this is the mom here, right? This is the grandfather. The grandfather had the typical males of dyskeratosis congenita, which in this case this shows an extreme version with disappearing male. He had bone marrow failure as a teenager and unfortunately died related to bone marrow failure. Okay. His daughter, the mom of our kids here, was completely healthy. She had no medical problems. Unfortunately, she had 3 affected boys who had classic dyskeratosis congenita. And what studying families like this led Indrajit Dokal, who's in the UK, and his team in the late 90s discovered the X-linked form of dyskeratosis congenita, which is variations in a gene called dyskerin or DKC1. And at the same time, another lab was studying telomere biology and found that patients that cells from people with this mutation actually had very short telomeres and low level of telomerase. So this was about all we knew about 20 years ago. At the same time, there were more and more reports in the literature showing it wasn't just males that were affected. And families with autosomal dominant and autosomal recessive inheritance were being reported in the literature. So then, and some people had no family history. So then around 2005, a couple other genes, TERT or telomerase and TERT, which is part of the enzyme complex that helps extend telomeres. That's what this is. The colored symbols here are part of the complex that makes those telomeres longer. So there were several reports, again, about 20 years ago, showing that actually mutations in TERT or TERT could cause a telomere biology disorder, which we weren't calling it at the time. I started working on this sort of right around then, 2004 or 5. And one of the first things I did as part of our inherited bone marrow failure syndrome study at the National Cancer Institute was actually, as we were enrolling patients with dyskeratosis congenita, we said, they don't have a support. The doctors and physicians aren't really collaborating. The patients and families aren't really talking. So we organized a workshop that we held in 2008 at the NIH, and it was amazing. We had about 40 patients or families there, and about 40 researchers and clinicians, and had the first workshop. This led to what became dyskeratosis congenita outreach around the same time, led by Nancy Cornelius, Robin Harris, and her husband Joe Carlson, and a few other people who really put this group together in a very foundational grassroots way. And some of you were part of the original group. And I like this. I found the letter. I was reminiscing and thinking about this, and for too long, families worldwide affected by dyskeratosis congenita have nowhere to turn for support. No more. A new volunteer organization, BC Outreach, has formed to help families and professionals reach out to one another. And that was really the whole point of this group, was connecting people. Our first camp, I can't believe it, was almost 15 years ago. In 2010, this is a photo of the first group, including a few doctors and patients and families. So that brings me, this is when we were talking about dyskeratosis congenita BC, which a lot of you, I'm sure, have heard about. But let's talk about the names and the nomenclature for a sec. So what's a name? If you look in the literature, all these names, and I'm going to run through them quite quickly in some other examples. But what's common to everything is abnormal, short, or dysfunctional telomeres. Okay. And this has led us, this definition that Dr. Bertucci and I worked together to define, along with more than 30 members of the telomere clinical and research basic science communities, where we're defining telomere biology disorders as a specific spectrum of illnesses caused by telomere dysfunction. The medical complications of patients with TBDs result from limited stem cell renewal, induced by critically short telomeres. And some TBDs though have telomere instability without having short telomeres. So this is our bone marrow failure syndrome study, and I have some information, and if anyone's interested, it's a long-term cohort study that we're doing to really continue to define the genetics, the clinical complications, and the underlying biology. And I'm happy to talk to folks about this more later. So one of the early things that we were able to do in that study by evaluating patients and families was to work with the lab that's now called Repeat Diagnostics to develop the diagnostic test for BC at the time. And this is where I wanted to also show, just to emphasize, the telomere length curves. Many of you have maybe seen this in yours or your kids' results. This is the first percentile for age. This is the 99th percentile. So if you all remember, when kids are young and growing, the pediatricians are focused on what percentile is their height and weight. And that's because there's normal variation in the height and weight, and there is in telomeres. So 99% of the general population is going to have telomeres that fall in this range, which is actually quite a wide range, okay? So what we showed was actually that the people with DC had much, much shorter telomeres than the general population. And over time, this led us and many others to discover additional genetic causes of the telomere biology disorders. In the last 20 years, we've gone from knowing 3 genes to at least 18 or 19, depending on definitions. And most of them we're finding more rare genes, but we're expanded the number of genes that we know, which has also really helped us understand the biology. Okay, because as you all know, these are complex disorders. Defining the problem is really the first step to figuring out how to fix it. Okay, so we have lots of genes now, which also makes genetic counseling also complicated and even more important. So I'll go through again at a high level a lot of the clinical features, because we're going to spend a lot of time tomorrow talking more about this. And I really want to just emphasize over and over again, we talk a lot of big scary things here. And a lot of you have been through, or your kids have been through big scary things. But remember, everyone is different. You're you, your kid is themselves. And so not everything we talk about is going to happen to everyone, okay? Or it's not going to be in the same way. We're learning more and more every day. And the talks that we gave here at the very first camp are very different than what we're giving now. And we're making a lot of major advances. So keep that in mind as you sort of think through and learn about all this, because I know it's overwhelming. So I've kind of been to the TBDs in different groups now. So dyskeratosis congenital, which is the classic TBD, is associated with abnormal nails, abnormal skin pigmentation, oral lipoplegia, white spots in the mouth. Patients with oral Richardson syndrome can present very early in infancy, and they often will have those features and immunodeficiency and cerebellar hypoplasia, which is the cerebellum is the back part in your brain that is important for coordination. Okay. Coates plus and Revenge syndromes are rare variants as well that are associated with bleeding due to some blood vessel abnormalities. These are people who usually present very early in life as children with really, really short telomeres shown here, much shorter than the first percentile. And when we look at what are the genes most likely to be affected by for these people, is they're usually going to be the X-linked recessive, so dysKaren, or the autosomal recessive. So that means they inherited an abnormal copy from mom and an abnormal copy of that gene from dad. And they present more earlier. And you can see we moved from just having one or two genes to many autosomal recessive genes. Now, kind of at almost the other end of the spectrum of the TBDs are the patients who actually present with only, quote unquote, one problem or maybe two, and maybe they don't present until they're 40, even 30, 40, 50. We've diagnosed people in their 60s where they may have aplastic anemia, which is another word for bone marrow failure, which this shown here, the red is the bone in the bone marrow, and that all that white emptiness is the lack of cells in there. We'll talk more about that. They may present with abnormalities in their blood cells, myelodysplastic syndrome. Head and neck squamous cell cancer is also, we've had some patients present with that as adults. Pulmonary fibrosis, we'll talk more about that again tomorrow, and then liver disease. When you look at these people, they're more likely to present as adults, although there's a lot of variability. But they're also more likely to have telomeres that are kind of in that 1st to 10th percentile for age. So they're short, but technically 10% of the population has telomeres at the 10th percentile. So that's where also understanding the family history and the genetics in those disorders is important, where we now have a host of different genes, and these are folks who are more likely to have autosomal dominant inheritance. So have inherited one abnormal gene from a parent. Okay, so there's a lot that's gone on, and I want to just point out that the work that the physician and scientist community is doing together in the Clinical Care Consortium of Telomere-Associated Ailments, which was started probably 15 years ago. And we've since done a lot of group studies where we're collecting data to better define what's the problem. Because in a rare disease, it's really important to define that problem so you can hopefully intervene. I think you all know, and I don't expect you to read any of this. This is from our yearly review by Dr. Bertucci and Dr. Revy, that living with a telomere biology disorder is complicated, right? We have lots of different body systems affected, lots of different ages, lots of specialists to coordinate. One of the things that we did in collaboration with Team Telomere is my team has a psychosocial research group, and the team interviewed 32 either adults with a TBD or adult caregiver of a person with a TBD. And this is the first time that in a sort of systematic scientific approach that we had really compiled data looking at what are the challenges you all face. You all know this, right? But it really helps the scientific and the clinical community to understand, okay, we need to offer, in this case, we need to offer more social work type support because care coordination is a huge challenge. thinking about psychological well-being, health well-being of folks as well is also incredibly important. So this is just a quick snapshot. We now have 22 institutions around the world who are participating in the consortium. We have several different studies ongoing, which has just been really exciting. And just one additional bit of history. So our first group, our first Telomere Biology Disorder guidelines was in 2015. And now I think you all should have at least an electronic copy of the guidelines, which was just came out a couple of years ago, and we really emphasize the importance of a medical home for everyone and talking with providers. And I know it's one thing when we get referrals, we all try to emphasize, we all need, you know, the docs need to really work hard to coordinate and build that environment for all the complexities. And I will end here with pictures of camp from across the years. And if we have time, I can take some questions, but regardless, I'll be around for the next 2 days.